Microbial taxonomy. Many different types of organisms are able to parasitize humans and animals. The correct identification of these organisms is important because of their impact on the health of humans and animals. Identification is based on the biological classification, the purpose of which is characterize, group, and name organisms in a manner that will promote accurate scientific communication. Classification is the order arrangement of the organisms into groups on the basis of their relationship to one another. Taxonomy is defined as the theoretical study of classification, includes its basis, principle, procedures, and rules. Nomenclature is the applying of names to the groups delineated in the classification steps. Identification is the allocation or assignment of an unidentified organism to the correct and named groups. All organisms can be grouped into a series of subdivisions that make the taxonomic hierarchy. For eukaryotic organisms, a species is a group of closely related organisms that breed among themselves. A genus consists of species that differ from each other in certain ways but are related by descent, just as a number of species make up a genus. Related genera make up a family. A group of similar families constitute an order, and a group of similar order make up a class. Related classes make up division. In zoology, the comparable term phylum is used. Thus, a particular organism has a genus name and specific epithet and belong to a family, order, class, phylum, or division. All divisions that are related to each other make up a kingdom. And we detail this in this literature. Here we find the scientific classification established by Carl Linnaeus. Each species has to be assigned to a genus binary nomenclature, which in turn is a lower level of hierarchy of ranks: family, kingdom, phylum, class, order, family, genus, species. In the currently accepted classification of life, there are three domains: eukaryote, bacteria, and archaea. Microbial taxonomy is a means by which microorganisms can be grouped together. Organisms having similarities with respect to the criteria used are in the same group and are separated from the other group of microorganisms that have different characteristics. What's taxonomy? Taxonomy is the science of classification of organisms with the goal of showing relationship among organisms. Bacterial taxonomy is the rank-based classification of bacteria. The aim of classification of to delineate group of organisms about which one can generalize knowledge obtained from studying few strains. Such a group is called a taxon. Taxonomy includes identification, nomenclature, classification. Identification is the process of studying and recording the identical and distinguishing features. Nomenclature is the process of assigning names to the various taxonomic ranking of each living organism. Classification is the orderly arrangement of organisms into groups, preferably in a format that shows the relationship. Basis of taxonomy: phonetic system, group organisms based on mutual similarity of phenotypic characteristic. Phylogenetic system, group organisms based on shared genetic heritage. Two methods: the shape of the microorganism and the genetic of the microorganism, and we will detail it. Taxonomy of bacteria. Living organisms were once divided into only two kingdoms, Plantae and Animalia. Previously, bacteria, fungi, algae were classified with plants. Protozoa were classified as animals. Haeckel in 1865 proposed third kingdom, Protista for unicellular microorganisms such as bacteria, algae, fungi, and protozoa. Bacteria were separated into the kingdom Prokaryote or Monera in 1969, only 1969, by Whitaker. He divided it into 5 kingdoms. Taxonomy of bacteria. Living organisms were once divided into only 2 kingdoms, Plantae and Animalia. Bacteria, fungi, and algae were classified as plants. Protozoa were classified as animals. Haeckel in 1865 proposed third kingdom protista for unicellular microorganisms such as bacteria, algae, fungi, and protozoa. Bacteria were separated into the kingdom Prokaryote or Monera in 1969 by Whitaker Kingdom classification based on cellular organization and nutritional pattern. Prokaryote, protista, mastiti or fungi, plantae, animalia. Animalia, eukaryote, unicellular, heterotrophic or ingestive animals, plantae, plants, also algae, mosses, ferns. Mycete, eukaryote, unicellular or multicellular, heterotrophic or absorptive. Protista, eukaryote, unicellular, colonial, heterotrophic, protozoa, similar mold, some algae. Prokaryote or Monera, prokaryote, unicellular, a variable nutrition pattern, bacteria. The three domain, currently Carl Weiss and George Fox in 1977. Currently living organisms are divided into three super kingdoms called as domains. Eucora, domain include plants, animal, fungi, and protists. Eubacteria, domain include bacteria with protidoglycan in cell wall. Archaea, domain include bacteria, archaea with unusual cell wall. Here the bacteria, here archaea and eukarya. Scientific nomenclature. According to scientific nomenclature, such as bacteria is assigned to name binomial nomenclature, genus and species, where the genus and the species rule, and they are in Latin, and this is not a changed language. Staph aureus, we say Staph aureus. Escherichia coli. Nobody can change this with language. It changes as language changes. The dictionaries are renewed every 10 years, every 5 years. The Latin is not changed language. We use it in the nomenclature. Laws for the assignment of names to bacteria are established by the International Committee on Systemic Bacteriology. Nomenclature of bacteria. According to scientific nomenclature, each bacteria is assigned two names: binomial nomenclature, a genus and a species. Criteria used for bacterial classification. Criteria used for bacterial classification: phenotypic character, morphological character, stained film, wet film, or stained film and examined under microscope, biochemical character, biochemical test, culture character, by cultivation on liquid or solid media, by cultivation on selective media, by cultivation on blood agar. Serological characters depend on somatic or flagellar or capsular antigen. The use of certain serological tests such as FAT, ELISA, and RIA. Classification depend on bacterial DNA. Genome size. Each bacteria has its DNA genome size, so used for classification. GC content. It depends on high advanced technology. It varies from 25 to 75 mole% for all bacteria. Micrococcus has high GC content, while Staphylococcus has low GC content. DNA relatedness depend upon DNA strand complementary to other strand from other microorganisms. One strand from E. coli can combine and anneal with other strand from Salmonella. Strand from E. coli can't anneal with another strand from Staphylococcus. DNA homology. It means that the nucleotide sequence is like that of other bacteria. Bergey's Manual of Determinative Bacteriology, International Standard for Bacterial Taxonomy, first published in 1923. 9 editions have been published from 1980. It's renamed as Bergey Manual for Systemic Bacteriology. Bergey's Manual of Systemic Bacteriology, published as 4 volumes from 1984 to 2012. 5 volume set. Currently, Bergey Manual of Systemic Bacteriology of Archaea and Bacteria. In 2015, one online book replaced the 5 volume set, and we depend on this manual on the taxonomy of bacteria. In original 4-volume of Bergey Manual of Systemic Bacteriology, bacteria were classified in 33 sections based on phenotypic, not phylogenetic characteristics, ranking of prokaryote, division, graciliputes, and firmicutes, and tenricutes, mendocicutes. The current 5-volume define taxa not in terms of phenotypic, but solely in 16 as phylogeny. Graciliputes, prokaryotes with a gram-negative cell wall. Firmicutes, thick skin prokaryotes with a gram-positive cell wall. Tenricutes, soft or tender skin prokaryotes that lack a cell wall. Mendocicutes, skin with folds prokaryotes with unusual cell wall.